Okay, now I would like to talk about history of cellulose. Cellulose was discovered in 1838 by the French chemist named Anselme Payen. Cellulose was used to produce the first successful thermoplastic polymer, celluloid hair manufacturing company in 1870. Production of rayon, the artificial silk from cellulose, began in the 1890s, and cellulose was invented in 1912. Hermes found between the polymer structure of cellulose in 1920. The compound was first chemically synthesized without the use of any biological by microbiology and soda.
Cellulose has no taste, is odorless, is hydrophilic with a contact angle of 20 to 30, is insoluble in water and most organic solvent, is chiral and is biodegradable. It can be broken down chemically into its glucose units by treating it with concentrated acids at high temperature. Cellulose is derived from D-glucose units, which condense through beta 1 until 4 glycosidic bonds. This linkage motif contrasts with that for alpha 1 until 4 glycosidic bonds present in starch, glycogen, and other carbohydrates. Cellulose is a straight-chain polymer. Unlike starch, no coiling or branching occurs, and the molecule adopts an extended and rather stiff rod-like conformation, aided by the equatorial conformation of the glucose residues. The multiple hydroxyl groups on the glucose from one chain from hydrogen bonds with oxygen atoms on the same or on a neighbor chain, holding the chains firmly together side by side and forming microfibers with high tensile strength. This confers tensile strength in cell walls, where cellulose microfibers are measured into a polysaccharide matrix. Compared to starch, cellulose is also much more crystalline, whereas starch undergoes a crystalline to amorphous transition when heated beyond 60 to 70 Celsius in water. The conversion of cellulose I to cellulose II is irreversible, suggesting that cellulose 1 is metastable and cellulose 2 is stable. With various chemical treatments, it is possible to produce a structure cellulose 3 and cellulose 4. Many properties of cellulose depend on its chain's length or degree of polymerization, the number of glucose units that make up one polymer molecule. Cellulose from wood pulp has typical chain length between 300 and 1,700 units. Cotton and other plant fibers as well as bacterial cellulose have chain lengths ranging from 800 to 10,000 units. Molecules with very small chain lengths resulting from the breakdown of cellulose are known as cellodextrins. In contrast to long-chain cellulose, cellodextrins are typically soluble in water and organic solvents. Plant-derived cellulose is usually found in a mixture with hemicellulose, lignin, pectin, and other substances. While bacterial cellulose is quite pure, has a much higher water content and higher tensile strength due to higher chain length. Cellulose is soluble in Skiweser reagents, CED, cordaxans, and methylmorpholin and oxide, and lithium fluoride, dimethylacetamide. This is used in the production of regenerated cellulose such as viscose and cellophane from dissolving pulp. Cellulose is also soluble in many kinds of ionic liquids. Cellulose consists of crystalline and amorphous regions. By treating it with strong acid, the amorphous regions can be broken up, thereby producing nanocrystalline cellulose, a novel material with many desirable properties. Recently, nanocrystalline cellulose was used as filler paste in bio-based polymer matrices to produce nanocomposites with superior thermal and mechanical properties. Hemicellulose is a polysaccharide related to cellulose that comprises about 20% of the biomass of most plants. In contrast to cellulose, hemicellulose is derived from several sugars in addition to glucose, especially xylose but also including mannose, galactose, rhamnose, and arabinose. Hemicellulose consists of shorter chains, around 200 sugar units. Furthermore, hemicellulose is branched, whereas cellulose is unbranched.
Biosynthesis. In vascular plants, cellulose is synthesized at the plasma membrane by rosette terminal complexes, RTCS. The RTCS are hexameric protein structures approximately 25 nanometers in diameter that contain the cellulose synthase enzymes that synthesize the individual cellulose chains. Each RTC floats in the cell's plasma membrane and spins a microfibril into the cell wall. RTCS contain at least 3 different cellulose synthases encoded by CESA genes in an unknown stoichiometry. Separate sets of CESA genes are involved in primary and secondary cell wall biosynthesis. Cellulose synthesis requires chain initiation and elongation, and the two processes are separate. CESA glucosyl transferase initiates cellulose polymerization using a steroid primer, cytosterol beta glucoside, and UDP glucose. Cellulose synthase utilizes UDP D-glucose precursors to elongate the growing cellulose chain. A cellulase may function to cleavage the primer from the mature chain. Cellulose is also synthesized by animals, particularly in the test of ascidians where the cellulose was intrinsically turned to niacin, although it is also a minor component of mammalian connective tissue.
Breakdown of cellulolysis. Cellulolysis is the process of breaking down cellulose into smaller polysaccharida called as cellulodextrin or completely into glucose unit. This is a hydrolysis reaction. Because cellulose molecules bind strongly to each other, cellulolysis is relatively difficult compared to the breakdown of other polysaccharides. However, this process can be significantly intensified in a proper solvent, for example, in an ionic liquid. Most mammals have very limited ability to digest dietary fibers such as cellulose. Cow and sheep contain certain symbiotic anaerobic bacteria, for example, Cellulomonas. And these bacteria produce enzymes called cellulases that help the microorganism to break down cellulose. The breakdown of these products are then used by the bacteria for proliferation. The bacterial mass is later digested in its digestive system, for example, stomach.
Hi guys, I'm going to talk about the uses of cellulose in pharmaceutical products. There are many types of uses in pharmaceutical products such as in suspension, emulsion. So, in suspension and emulsion, the cellulose play an important role such as thickening agent and gelling agent and giving a good taste for it. Hi guys, so this is the example of product that use cellulose in pharmaceutical. Thank you.
What is cellulose and how it is used to make ethanol? When plants grow, they synthesize a variety of different compounds called carbohydrate. There are many different plants stored carbohydrates. For example, sugar, which is found in fruit, starch, which is found in potatoes, enzymes are found in leaf and stems. Every plant from grasses to trees has cellulose. It is most bounded organic compound in the earth. Cellulose is the part of cell wall which give plants structure and protection. Cellulose contain high energy sugar called glucose, which could be broken down to fluid like ethanol. This ethanol could be used to place gas in cars. Thus breaking down the plant cellulose to sugar, it seems too simple enough.
So why scientists have difficult transforming cellulose to ethanol? To understand this problem, the scientists start looking at structure of cellulose. Cellulose is actually mode building block of glucose, simple sugar. In plants, these building blocks are only together in long chain inside cell walls. These glucose chains are found in complex where other sugar like hemicellulose and structure materials called lignin. This is where protect and support high energy sugars. Because of cellulose are tangled, hemicellulose and lignin is different to separate out of the plant material for conversion to ethanol.
So, how scientists solve this problem and turn this tangled hemicellulose to ethanol? First, plants material are chopped down into small pieces at the same size. Then, scientists must detangle the plant material in a process called pre-treatment. It is a heat or chemical process. This exposed plant cell wall loses the cellulose from the lignin and hemicellulose. After pretreatment, enzymes are used to chop the cellulose in a chain of glucose to individual sugar. Next, the glucose sugar are fermented in a large tank by microbes like E. coli and yeast. These microbes eat sugars and release ethanol as a waste product. The ethanol product is recovered inside the gas station using vesicles. Right now, the step costly and consuming time. But scientists are working to make the whole process less expensive, so one day ethanol produced from cellulose can roll the road. Thank you.
Cintaku padanya, walaupun sengsara, namun ku masih sayang, namun ku tiada, namun ku